sequences or amino acid sequences that make this immunoglobulin. And we can also see there is a presence of carbohydrates. So now let's look at the detailed structure of this immunoglobulins. Now if you look at the structure, it is made up with several different parts. If this is a Y-shaped molecule, it is having different parts, like say this is the upper region. So let me consider it from the beginning, that there it is having two different color indicating two different type of chain. This green or light green color, this chain is called the heavy chain because it is long, it is heavy. And these blue colored segments are very less, very short and small, so they are called the light chain. So it is consisting of two heavy chains, and also two light chains that we can see in this picture. And on this structure what we are having, there are regions, like here it is the region. This part of the region of this top of the Y is the region for antigen binding. So that's why it is termed as AG binding or antigen binding region. And obviously at the bottom part what we can see, this is the region, this part of the region is called the FC region, and this is called the binding of receptor region, because using this bottom part. So let me change the color. So here it is. So using this bottom part, it actually binds with different receptor molecules onto the surface of different types of cell inside our body. Okay. And in this FC region there are two different parts. One is a part for placental transfer, another one is a part for the complement binding site. So here it is. So what we can see, not only two different chains, one heavy and one light, but also what we can see, we are see this kind of hinge like structures like this, right? So this is a typical kind of protein folding that has been observed for this kind of immunoglobulins. The typical kind of folding, this folding is, as you can see, throughout the body it is having this kind of folding. Okay. And this folding is accomplished due to the presence of disulfide linkages. Now here we go. If this red thing which I have drawn here, it is a heavy chain, for example. Now if it is a chain, so it is previously say like this straight chain, so it's a long straight chain like that. Now once this straight chain is has been folded in this way to get a structure with this kind of loops. Now how can they get the structure? They get the structure using this disulfide linkage, as I am drawing here with this blue color. Now these blue color regions here are the disulfide linkages. So here is the sulfur, here is another sulfur, so they are linked each other by the disulfide linkage, so that these two places, suppose this is a sulfur, this is another sulfur, they will make a loop using this disulfide linkage, so that they get this kind of loop like structure. So what we get in both these cases, we are having several loop like structure in both these cases, on heavy chain as well as on light chain. So this kind of linkage, when you're having the linkage between the same amino acid chain or polypeptide chain, is called intra-chain. It's called intra-chain sulfide bonding. Intra, because it is between a same polypeptide chain. Now if I look that when all these things are arranged, now this heavy chain is totally arranged. Now similarly the light chain is also made this kind of loop structure. Now there is something which is holding this heavy chain and light chain together, right? Here we can see this red region here, and also this region. These two are the regions, again another type of disulfide linkage, which are holding one heavy chain with another light chain. Now this disulfide linkage, as it links two different types of polypeptide chains together, it is called interchain disulfide linkage. Interchain disulfide linkage. Okay. So here it is, two different types of intrachain as well as the interchain disulfide linkage that we can see in this picture. Okay. Now if we look at this detailed picture, what we can get from here it is the antigen binding as I've seen, and here it is the FC receptor, and so complement binding. So this is called the biological activity domain, and here it are the regions where the actual uh, carbohydrates are bound. So if we consider the structure here, two loops on the top, two loops on the bottom in the FC part, and the top part is called FAB part. Why they are called FAB and FC? This FAB part is responsible for antigen binding, that's why it's termed as antigen binding or FAB, and C is the part for the cellular receptors, so that's why it is termed as FC part, which is the bottom part. That means this one, this whole part is FC part. Okay. Now two loops in the FC part, two loops in the FAB part in both these cases. So the second loop of on this FAB part is not the first loop of this FC part is bound with this carbohydrate structures. Okay, as you can see in both these cases, the carbohydrate structures are bound. Okay, now in this picture it is much more clear how the disulfide linkages are there. So here are the disulfide linkage, here the looping is not shown,